is genetic engineering. So genetic engineering, or also called genetic modification, is the process of altering an organism's DNA in a laboratory to change its characteristics, such as by adding, deleting, or changing a specific gene. So for example, genetic engineering may involve adding a gene from one species to an organism from a different species to produce a desired trait. Used in research and industry, genetic engineering has been applied to the productions of cancer therapies, brewing yeast, genetically modified plants and livestock, and more.
Hi everyone, I'm Raymond, and I'll be your next presenter. Let's explore the key differences between genetic engineering, conventional breeding, and general biotechnology applications. Genetic engineering stands out due to its precision, efficiency, and the specific results it can achieve. We'll discuss how these differences influence various applications and why genetic engineering is becoming a preferred method in certain situations. Understanding these distinctions will provide a clearer view of the benefits and the possibilities of genetic engineering compared to more traditional approaches. Traditional breeding involves carefully selecting organisms and crossing them over multiple generations. This process aims to combine desirable traits from different parent organisms. One key characteristic is that trait inheritance is random and less precise. This means that breeders have limited control over which traits are passed on. Both desirable and undesirable traits can be transferred together, requiring further selection to eliminate unwanted characteristics. Conventional breeding is limited to sexually compatible species, restricting the range of genetic diversity that can be introduced. The development process is slow and time consuming, often requiring many years to achieve the desired results. In conventional breeding, you start with a virus resistant plant and a high yield crop. These are crossbreed, and over time, we aim to combine both traits into a single plant. In genetic modification, we also start with a virus resistant plant and a high yield crop. However, instead of crossbreeding, we directly modify the genetic material to insert the desired traits. And the outcome, both methods can achieve the same result, a virus resistant and high yield crop. However, genetic modification offers a more direct and precise route. As you can see, while both approaches can lead to improved crops, genetic modification offers a more targeted and efficient means of achieving specific traits. Let's examine genetic engineering, aka biotechnology tool. It involves direct DNA modification at the molecular level. Specific genes are inserted, removed, or altered for targeted trait introduction. Allows precise and predictable trait expression. Enables gene transfer between unrelated species. Results in faster development of improved organisms. In summary, genetic engineering offers superior control and efficiency in organism modification. Let's place genetic engineering in the context of biotechnology. Biotechnology is a broad field using biological processes like fermentation and tissue culture. Traditional biotechnology doesn't directly alter DNA, while genetic engineering is an advanced branch that does. Genetic engineering focuses on gene level manipulation for targeted improvements. In summary, biotechnology includes many techniques, with genetic engineering offering a precise, modern approach to organism modification. Now let's turn our attention to the core techniques involved in genetic engineering. Recombinant DNA is an artificially made DNA strand formed by combining two or more gene sequences. It is constructed artificially, meaning it doesn't occur naturally, but is created in a laboratory setting. Recombinant DNA technology is essential for creating organisms with new or enhanced traits. Let's take a moment to acknowledge the pioneers of recombinant DNA technology. Recombinant DNA technology is a recent advancement in biotechnology, developed by scientists Boyer and Cohen in 1973. As shown in the slide, Stanley and Cohen received the Nobel Prize in Medicine in 1986 for his work on discoveries of growth factors. Stanley and Cohen and Herbert Boyer together constructed the first recombinant DNA using bacterial DNA and plasmids. Their groundbreaking work laid the foundation of modern genetic engineering. Now let's delve into the process of how recombinant DNA is created. Six key steps involved in creating recombinant DNA. Number 1. Isolating. This step involves extracting and purifying the target gene, the DNA you want to clone, and the vector, a carrier like a plasmid. Number 2. Cutting or cleavage. Restriction enzymes, also called molecular scissors, are used to cut both the target DNA and the vector DNA at specific sites. Number 3. Joining or ligation. The cut target gene is inserted into the cut vector using the enzyme DNA ligase. DNA ligase helps form a phosphodiester bond between the vector and insert. Number 4. Transforming. This step introduces the recombinant DNA into a host cell. The host cell takes up the recombinant DNA and allows it to replicate. Number 5. Cloning. Once inside the host, the recombinant DNA is copied at the host cell divides, creating many identical copies. Number 6. Selecting or screening. Identifying host cells that have successfully taken up the recombinant DNA molecule is crucial. It is important to identify and isolate the cells that have successfully incorporated the recombinant DNA. These steps ensure the recombinant DNA is replicated and the desired cells are isolated for further use. Now let's focus on CRISPR-Cas9, a powerful tool for gene editing. CRISPR stands for Clustered Regularly Interspersed Short Palindromic Repeats. It's a unique technology that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. CRISPR is derived from a natural defense system in bacteria and archaea, used to protect against invading viruses. This system has been adapted and refined for precise gene editing in various organisms. Let's explore the mechanics of how CRISPR-Cas9 works at the molecular level. The CRISPR-Cas9 system has two key components. Number 1. Cas9 or CRISPR-associated protein 9. Cas9 is an enzyme that acts as a pair of molecular scissors that can cut the two strands of DNA at a specific location in the genome, allowing bits of DNA to be added or removed. Number 2. Guide RNA or gRNA. A specific RNA sequence that recognizes the region of interest in the target DNA. It binds with the Cas9 protein and directs it to the target site to perform the modification process. The gRNA guides the Cas9 enzyme to the exact location in the DNA where a cut needs to be made, enabling precise gene editing. CRISPR-Cas9 is like a precise pair of molecular scissors that can edit DNA. Here's how it works. Number 1. Finding the target. Imagine DNA as a long instruction manual. The first step is to identify the specific sentence or gene you want to change. Scientists design a guide RNA or gRNA that matches this sentence. Number 2. Guiding the scissors. The gRNA acts like a GPS, leading the Cas9 enzyme to the exact location on the DNA. The gRNA and Cas9 form a complex ready to make a cut. Number 3. Cutting the DNA. The Cas9 enzyme is the scissors. It waits for the gRNA to guide it to the right spot. Once there, Cas9 makes a precise double-strand break in the DNA. Number 4. Repairing the cut. After the cut, the cell's natural repair mechanisms jump into action. There are few possible outcomes: mutation or the error. The cell might try to fix the break but makes a mistake, leading to a disruption or inactivation of the gene, deletion or removal. Scientists can engineer the system to completely remove the gene, insertion, or replacement. Scientists can provide a new piece of DNA, which the cell inserts into the break, replacing the original gene with a new one. In essence, CRISPR-Cas9 allows us to precisely target, cut, and modify DNA sequences, offering powerful possibilities for gene editing. When CRISPR-Cas9 cuts DNA, the cell's natural repair mechanisms kick in. Let's explore these pathways. NHEJ, or non-homologous end joining. NHEJ is an error-prone DNA repair pathway that rejoins broken DNA ends without requiring a template with homologous sequences. This mechanism often leads to small insertions or deletions. HDR, homology directed repair. HDR allows for precise editing of the genome by including a repair template in the process. Using a single-stranded template ensures that this template is not randomly inserted elsewhere in the genome or targeted by the Cas9 enzyme. Understanding these repair pathways is crucial for controlling the outcome of CRISPR-Cas9 gene editing.
Gene silencing. Gene silencing is defined as the prevention of gene expression which can occur through mechanisms that spread from silencer elements and can affect extensive regions of DNA. Gene silencing is one of the most efficient tools which downregulates the expressions of a gene in a definite way and has compelling influence on crop improvements. So gene silencing can be accomplished in transcriptional and post-transcriptional stages. From occurs through the refinement of the process of transcriptions, while later can occur through mRNA degradation. We have two mechanisms of gene silencing, which is the transcriptional gene silencing. This prevents a gene from being transcribed into mRNA in the first place. Second is the post-transcriptional gene silencing. This stops the gene from being translated into the protein after the mRNA has already been created. Transgenesis. Transgenesis defined as the biotechnological methods for producing genetically modified animals utilizing techniques such as sperm-mediated gene transfer to introduce exogenous DNA into oxide for application in veterinary medicines, biomedicines, and agriculture. So, transgenesis refers to the process of introducing an exogenous or modified genes or the transgenes into a recipient organisms of the same or different species from which the gene is derived. The transgenes become incorporated to the genome of the host organisms and can be transmitted to the offspring.
So, I have a trivia question for all of you. Which technology allows scientists to precisely edit genes often compared to using molecular scissors? A. Selective breeding. B. Hybridization. C. CRISPR-Cas9. And D. Cloning. So what's your answer? And the answer is C. CRISPR-Cas9.
Good day, everyone. To formally begin my discussion, allow me to introduce to you myself. I am T. Miraflor, a forestry student. What is genetic engineering in agriculture means? Genetic engineering in agriculture involves directly modifying a plant's DNA to introduce directly traits like pest growth, resistance, improvement, nutrients like golden rice or a higher yields, creating genetically modified organism for more resistance and productivity crops, reducing pesticides, herbicides needs, and adapting to climate change challenges, extreme temperature. Crops improvement. Pest resistance brought to tolerant enhancement. Information. Crops like corns that are resistant to diseases caused by fungi, bacteria, and viruses can reduce the needs of chemical pesticides. Biofortification, golden rice, and formation. Food crops provide a sustainable long-term strategy for delivering micronutrients, including vitamins A, brought its processor, B, carotene, to population in developing countries. Post-harvest and processing trades, delaying repaying important self-life enhancement, flavoring or texture, and formation. As a result, texturized vegetables, protein, are gaining consumers' attention and providing better options for a vegetarian. Students have found that the presentation of a texturized vegetables promoting regardless for whether soybeans protein is processed at low or high moisture levels relies on a combination of hydrophobic interaction, hydrogen bonds, disulfide bonds, and their synergistic interaction.
Application of genetic engineering in agriculture. Presenting the importance of application of biotechnology in modern agriculture. These are soil and environmental management. And this innovation aim to improve productivity, sustainability, and environmental protection, which are crucial in addressing global food security and environmental challenges. It highlights soil and environmental management as key application where biotechnology is used. This application help farmers improve crop growth, maintains healthy soil, and reduce harm to the environment. Overall, the goal is to make agriculture more productive, sustainable, and capable of addressing global food security and environmental problems. So first, soil and environmental management. One major contribution of biotechnology to soil and environmental management is the development of crops engineered for phytoremediation and nitrogen fixation. It explains that this biotechnological innovation help increase productivity, promote sustainability, and protect the environment, which are essential in addressing global food security and ongoing environmental challenges. Phytoremediation refers to the use of genetically modified plants to clean up contaminated soils. These crops are engineered to absorb, break down, or neutralize pollutants such as heavy metal, pesticides, and industrial toxins. And also, phytoremediation by doing this helps restore contaminated land and makes the environment safe for all. Another innovation is genetically engineered crops for improved nitrogen fixation. Normally, plants depend on nitrogen fertilizers to grow, which can be expensive and harmful to the environmental when overused. Through fixed nitrogen from the air or work more efficiently with nitrogen fixing bacteria in the soil. This reduces the need for chemical fertilizers, lower pollutants, cuts farm cost and support. Another function of nitrogen fixation are soil enrichment. Nitrogen fixing plants like legumes can improve soil fertility. Second is ecosystem balance. Nitrogen fixation helps maintain the balance of ecosystem, supporting biodiversity and ecosystem health. Third, the climate regulation. Nitrogen fixing plants can help and reduce greenhouse gas emission. And lastly, water quality. By reducing the need for synthetic fertilizer, nitrogen fixation can help prevent water pollution and maintain water quality. The other one is livestock and feed innovation. Biotechnology also plays a vital role in livestock production, particularly through the development of genetically modified feed crops. GM feed crops are engineered to have improved digestibility and enhanced nutritional content, leading to a healthier livestock, improved growth, and more efficient meat and milk production. For example, GM crops and soybeans may contain higher protein levels or modified starches that are easier for animals to digest, allowing animals to absorb nutrients more efficiently, grow healthier, and produce better meat, milk, or egg. Biotechnology innovation in soil management and livestock feed play a significant role in creating a more sustainable and productive agricultural system. Crops engineered for phytoremediation and nitrogen fixation help restore soil health and protect the environment. The benefits of soil management is microbial inoculants. Beneficial microorganisms like rhizobacteria and mycorrhizal fungi are used to improve soil health and increase nutrient uptake. The other one is biofertilizer. Natural fertilizer made from microorganisms, plants, or animals improve soil fertility. And also in livestock feed production is probiotics. The beneficial of probiotics in microorganisms added also to feed improve gut health and boost immunity. Second is enzymes. Feed additives like breakdown of complex nutrients. And also, lastly, is genetically modified. Crops engineered to produce specific nutrients or traits improve animal and productivity.
Case studies and local relevance. Biotechnology has a significant role in helping the Philippines combat the problems it encounters in its agriculture sector, including low crop productivity, pest, malnutrition, and climate change. Various biotechnology programs and projects that can be benefit the farmers and the Filipino people have been launched inside the Philippines. Some of the most prominent of these are BT eggplant, golden rice, and biotech corn. These are developed by major institutions such as Philippines and the biotech program of the Department of Agriculture as well as IRRI. BT eggplant. BT eggplant is among the first genetically modified food crops developed for the specific benefit of Filipino farmers. It is a resistant to the eggplant fruit and shoot borer, a common pest of eggplant, and a possible reason why eggplant is expensive, considering that to protect their harvest, farmers need to spray pesticides on a frequent basis, which is dangerous to their health and the planet too. In the case of BT eggplant, the plant is able to withstand the pest and as such, there is no need for chemical pesticides. Consequently, there is lower production cost and increased yield, as well as healthier working conditions for the farmer worker. Furthermore, the BT eggplant assists in the promotion of sustainable agriculture as it reduces environmental pollution. Golden rice is genetically modified to have high levels of beta-carotene, which can be converted into vitamin A. Vitamin A deficiency is an important public health concern in the Philippines, as it predisposes one to lose of vision and reduce immunity, particularly among children and pregnant women. The reason why golden rice was created was to address the problem of malnutrition among Filipinos by providing an additional source of vitamin A in the form of a staple food that Filipinos usually consume every day. Although it won't solve the problem of malnutrition entirely. There are other approaches that can be used in conjunction with golden rice to address the problem of malnutrition in the Philippines. Biotic corn, particularly BT corn, also gained acceptance among Filipino farmers. Biotic corn is modified so it will be resistant to certain insects like the Asian corn bug. Findings show that Filipino farmers growing biotech corn have increased crops and revenues notwithstanding their lower pesticide use. Moreover, corn is a significant crop based its use in foods, feeds, and industrial processes. Hence, biotic corn is a significant contributor to food security and rural development in the Philippines. Additionally, the success of biotic corn has contributed to raising the acceptance of biotechnology among farmers. Role of key institutions. PhilRice, Philippine Rice Research Institution, is involved in rice R&D, especially in improved varieties of rice and golden rice development. The Agricultural Department Biotech Program is engaged in promoting the proper usage of biotechnology and is working toward the safety of humanity and the environment. IRRI, International Rice Research Institute, is playing an important role in breeding better quality rice and scientific research that helps many Filipino farmers and consumers. Concluding, biotechnology projects like BT eggplant, golden rice, and biotic corn prove how scientific research can help tackle practical issues in Philippine agriculture and food security. Thanks to the help of organizations like PhilRice, DA, and IRRI, biotechnology research keeps on bringing promising developments.
Global innovation, virus-resistant papaya, drought-tolerant maize, and non-browning apple. So, global innovation means using science and technology to improve crops so they can solve real-world problems like plant disease, drought, and food waste. So, example include is virus-resistant papaya, drought-tolerant maize, and non-browning apples. This innovation help farmers, consumers, and food security worldwide. Virus-resistant papaya. A genetically modified papaya plant known as virus-resistant papaya is shielded from the papaya ring spot virus, which may decimate papaya crops. In a manner akin to that of a vaccine, scientists inserted a tiny fragment of the virus genetic material into the papaya to assist the plant identify and feed off the infection. This enables the papaya to develop properly and yield wholesome fruit even in a region where the virus is prevalent. The papaya ring spot virus, PRSV, was seriously harming papaya crops in Hawaii. So the researcher created and tested genetically altered papaya plants that are resistant to PRSV. The papaya plant was able to identify and protect itself from PRSV infection, thanks to the insertion of gene from virus. Consequently, the transgenic papaya maintained normal development and fruit quality, while exhibiting significant resistance to the virus. This innovation proved the value of genetic engineering in crop protection, prevented Hawaii's papaya industry from collapsing, and became one of the most well-known instances of biotechnology being applied to agriculture issue in real life. So, virus-resistant papaya is genetically modified papaya defined to fight the papaya ring spot virus, PRSV. Scientists inserted a small piece of the virus genetic material into the papaya plant. This plant recognizes and defends itself against the virus. As a result, papaya plant can grow healthy and produce good fruit, even the area where the virus is common. This saved the papaya industry in places like Hawaii. Drought-tolerant maize, a variety of maize corn that has been bred or genetically altered to survive and grow well even when there is minimal rainfall, is known as drought-tolerant maize. It can help farmers produce food during drought because, in contrast to conventional maize, can tolerate dry circumstances. This cultivar has been created to improve food security, particularly in areas vulnerable to water scarcity, and to assist farmers in adapting to climate change through initiatives like the one headed by CGIAR. So, drought-tolerant maize is a type of corn that can grow even with less water. It is specially developed to survive dry conditions better than regular corn. The Drought-Tolerant Maize Project was created and overseen by the CGER Group. They develop maize cultivar that can thrive on less water while still yielding high yields through their research and partnership with farmers. Their effort enhance food security and assist farmers in drought-prone regions in adapting to climate change. So, these maize help farmers in dry areas avoid crop loss. It improves food supply and help farmers adapt to climate change by producing crops even during drought. Non-browning apple, an apple that has undergone genetic editing or modification to prevent it from rapidly becoming brown. When cut, bruised, or exposed to air is known as non-browning apple. Polyphenol oxide, an enzyme that normally causes apple to brown. It's either decreased or inactive in non-browning apple. These apples are easier to use in salad, snack, or pre-cut fruit packs, and they maintain their fresh appearance for longer. They also contribute as a decrease in food waste. One well-known apple that does not brown is Arctic. So, non-browning apple are apples that do not turn brown quickly after being cut. They are modified to slow down the browning process, making them look fresh and longer. Traditional versus Arctic apple. Traditional apple turn brown after being cut because an enzyme reacts with oxygen, An Arctic apple are genetically modified to silence the gene responsible for this enzyme. So, this means Arctic apples stay white and fresh longer even after slicing. According to the article, the FDA has approved Arctic Gala apple, enabling their sale in the US. These apples are kind of genetically modified apple that help save food waste and prolong the fruit freshness by preventing browning when chopped or crushed. This certification is a significant step toward bringing new, consumer-friendly apple varieties to market. So, the FDA approved Arctic apple, meaning they are safe to eat. These apples help produce food waste and keep fruit fresh for a longer period. Their approval support the development of consumer-friendly and innovative food products.
Hi everyone, my name is Dayot Anthony Jr. And today I will discuss for a short topic about ethical, environment, and policy consideration of genetic engineering and its application in ethical consideration. Number 1, safety. Safety I means kaligtasan. Safety ensuring that genetically modified organism GMOs are safe for human health. Ay pagtiyak na ang mga genetically modified organism GMOs ay ligtas para sa kalusugan ng tao. Number 2, equity and access. Required right to access GMO. Pagpantay na karapatan para sa mga tao o para sa access ng GMO. Number 3, human intervention and informing consent. Informing consent to human, individual, and discussion. May kaalaman, pahintulot sa individual at talakayan ng mga tao. Ethical environment consideration. Gene flow. Gene flow is about the plants, ilang gene, and impact on non-target organism is makita to diyan ay bata or animals or plant. Biodiversity is about biodiversity, diyan nato makita ang mga healthy animal or biodiversity. Resistant. Resistant is the good for humans or the plants. Policy consideration. Regulation and approval. Government need clean and robust regulator teamwork to assist the safety of GMO crops for both human and the environment of regulate adhering to the precautionary principle. Policy consideration. Limitly, transparency is the crucial for public acceptance. Policy like National Bioengineer Food Disclosure Standard in the use required limitly to inform consuming about the percent of bioengineer and ingredient. Intellectual properties. Patent of GM seeds can put similar farmers at a disadvantage, raising issue of fair access and control over the food supply. Public perception. Public acceptance is a significant handle breeding the gap between scientific conception and public concern. Required transparent community and addressing public affairs. Global standard. International organizations like the PEO and WHO are involved in development standard for the safety assessment of GM foods, but global policy remaining a point of contention.
Ethical, environmental, and policy consideration are three interconnected areas to guide decision and action to ensure they are responsible, sustainable, and aligned with societal values. Here is the breakdown. Ethical consideration. These focus on moral principles and questions of right and wrong. They address how action affect people, communities, and future generations, including issues like respect for life, protecting biodiversity, and animal welfare. Environmental consideration. These center on natural world and that impact of action on ecosystem, resources, and planetary health. Key areas include resource conservation, reducing waste using renewable energy, managing water, land, sustainability. Policy consideration. These involve rules, laws, and guidelines that shape how society address ethical and environmental issues. They include developing and enforcing regulation, emission standard, protected areas designation. Together, these three areas work to ensure that decisions are not only practical or profitable but also just sustainable and supported by clear framework. Thank you.
Good day, everyone. As we come to the end of our presentation, let us take a moment to recap the key insights we have discussed about genetic engineering and its role in modern science and agriculture. First, we learned that genetic engineering is the direct modification of an organism DNA at the molecular level. Unlike conventional breeding, which relies on random inheritance over many generations, genetic engineering allows scientists to insert, remove, or alter specific genes with precision. This makes the process faster, more predictable, and more effective. We also explore recombinant DNA technology, which involves combining DNA from different biological sources to create new genetic combination. This technique has been fundamental in biotechnology, allowing the production of beneficial traits such as disease resistance, improved yield, and enhanced nutritional value in crops. Major focus of our discussion was CRISPR-Cas9 technology. CRISPR as high accurate gene editing tools where the guide RNA identifies a target DNA sequence and the Cas9 enzyme cuts the DNA at the exact location. This enables scientists to correct genetic errors, insert new genes, or disable harmful ones. Because of its precision and efficiency, CRISPR has evaluated genetic search and opened new responsibilities in medicine and agriculture.
In terms of agricultural application, genetic engineering plays a vital role in addressing global challenges. Genetically modified crops can be engineered for pest resistance, drought tolerance, and improved nutritional content. Example, such as biofortified crops demonstrates how biotechnology can help combat malnutrition and enhance food security, especially in developing regions. However, we also emphasize that scientific advancement must be guided by responsibility, ethically considerations, environmental sustainability, and clear policy framework are essential. This ensures that genetic technologies are not used solely or for profit or convenience, but in ways that are fair, safe, sustainable, and beneficial to society as whole. When these three areas work together, they help build public trust and ensure long-term success. At this point, we would like to express our sincere appreciation to everyone who participated in today's presentation.
Thank you for your time, attention, and engagement. Your willingness to listen, reflect, and ask questions made this discussion more meaningful and productive.
Formally recognize your participation. Certificates of participation will now be distributed. This certificate serves as appreciation for your involvement and interest in understanding genetic engineering and its real-world application.
Once again, thank you all for being part of this session. We hope this presentation has increased your knowledge, encouraged critical thinking, and inspired continued interest in biotechnology and genetic engineering.
Thank you and have a great day.